Welcome back, everyone. We're going to move on now to the next session, which is session 2A, activities of potential interest to the AMR therapeutics research community. And for that, I'm going to introduce again Jessica Bonham, who's head of antimicrobial resistance at MRC UK and chair of the JPIAMR industry working group and the moderator for this session. Jessica, the floor is yours.
It is my very great pleasure to be able to introduce this session, which is selected talks, 10-minute talks from abstracts which were submitted in the open call for research abstracts. These 4 talks have less in the way of research outputs and more in the way of support activities for research. And so we've set up this additional session, hence session 2A, which is really activities to support AMR therapeutics research in the community. And I would like to open the session by asking Jessica Lee from the Cystic Fibrosis Syndicate in AMR is the name of her talk, and she's from the Medicines Discovery Catapult. Thank you.
Thank you, Jess, and hello, thank you for inviting me to speak. I'll be talking today about the Cystic Fibrosis Syndicate in Antimicrobial Resistance, which is a partnership between the Medicines Discovery Catapult and the Cystic Fibrosis Trust. So CF is a genetic condition that causes the buildup of a thick, sticky mucus in the internal organs. This basically creates the perfect breeding ground for bacteria, fungi, and viruses. So people with CF have chronic infections within their lungs. So there's a huge unmet need here because people with CF die from antibiotic failures and new treatments are urgently needed. So some of the key CF pathogens are included in the WHO priority pathogen list. So Pseudomonas aeruginosa infects around 70% of people with CF in the UK and is in the priority 1 critical category. And CF represents an ideal model for other respiratory and AMR conditions. So we have a really good understanding of the CF disease biology. There are key assets and knowledge in UK academia and a growing SME community. And the CF Trust is invested heavily in developing research tools for the community. This includes a patient registry and a clinical trials accelerator platform. So coming back to this unmet need, we've set up the Cystic Fibrosis Syndicate in Antimicrobial Resistance, which aims to accelerate the translation of new CF antimicrobial treatments to the clinic through collaboration so that we can ultimately bring effective treatments to people with CF faster. And this is what the syndicate looks like. So the managing partners of the syndicate are the Medicines Discovery Catapult, which is a not-for-profit organisation aiming to reshape medicines discovery in the UK, and the Cystic Fibrosis Trust, which is the leading medical research charity funding cystic fibrosis research in the UK. We've also brought together industry with expertise in the area, so from the UK, France and Switzerland. We've brought together UK expertise from academia in CF and pulmonary infection. And on a project by project basis, we pull in clinical collaborators so we can get the right insight and expertise we need. And importantly, we involve people with CF in everything we do. So they sit on our steering committee and we involve them in individual projects through the syndicate. So you can see that we have a critical mass of cross-sector expertise here, and we're working together on a shared research agenda to accelerate the translation of CF antimicrobials to the clinic. So I don't know if everybody can see this little box here, I'm trying to remove it, I'm sorry. When we kicked off the syndicate about 1.5 years ago, we did some extensive landscape mapping work. We consulted with industry leaders and academic leaders, and we identified that despite the fact that there is some global activity in CF antimicrobial development, there are key challenges that remain. So one of those key challenges is the fact that there's no agreed preclinical screening and testing pathway So it's difficult for innovators in the area to know which preclinical assays and models they should use, and there's a lack of guidance from regulators in the space. It's also difficult for innovators to access the clinical samples and data they need for preclinical studies in CF. So what we've done as a syndicate is really try to create a toolkit so that we can support innovators in this space. So this is just a few examples of the kind of resources that we're developing for the community. They include guidance documents on which strains and isolates should be used in preclinical studies, and a biorepository to facilitate access to those samples. It includes a grant to map that preclinical space in CF, and CF infection target product profiles, and also different technologies and tools that can be used to accelerate progress in this area. And what I'm going to do for the rest of the talk is just to talk you through two of these different tools that we've been developing. So first of all, the CIHR biorepository, which is led by my colleague Beth. The vision here really is to build and strengthen access to high quality, clinically relevant samples, data and expertise. So we have a focus on strains and isolates, sputum samples, but also the facilities to undertake screening. And this isn't going to be a physical biorepository, but it's actually a virtual biorepository, which will enable us to leverage existing collections and expertise across the UK and provide that coordinating role. So innovators who want to access tissue samples can come to us, we can support them to develop a tissue access request, and then we can signpost and link them up with centres that can fulfil that request. And this is a 3-year programme. Each year it gets a bit more sophisticated, and the vision is that eventually it will lead to a sustainable biorepository. And we've also included the wishes of people with CF within this model. So we're exploring ways that we can notify donors of samples when their sample is being used. And this is to promote patient engagement and show people with CF that it does actually make a difference to donate their samples. So this biorepository, the pilot, has been funded by the Cystic Fibrosis Trust. We're right at the start of this pilot phase now, so hopefully by the summer we'll be launching the initial offer and beginning to help innovators to access samples and data. Another tool that we've been developing are patient-focused target product profiles, or TPPs, for CF-related infections. So target product profiles are tools that are used to really identify the requirements that a drug or a diagnostic needs to meet in order to be successful in the clinic. So for drugs, this includes things like the patient population, treatment duration, safety and toxicology requirements. And it's usually focused around a specific drug or drugs. What we've tried to do is to kind of tip that on its head a bit and actually focus the TPPs around the needs and priorities of people with CF. Because we think that if we can do this, we can start to guide and catalyse the discovery and development of new treatments that meet the needs and priorities of people with CF. So just to give you an idea of how we've gone about developing these TPPs, we started with a focus group where we brought together people with CF to understand what they want to see from new drugs in this area. And one theme, as an example, that came out is the fact that people with CF have a really high burden of care. So they take oral medications, nebulized medications, sometimes IV medications. They have demanding nutritional and physiotherapy regimes, and any new medicine needs to fit really easily into those regimes in order to maximize adherence. And themes like this run throughout the target product profiles. We then reinforce this with clinical and industry insights in order to create the TPP that you can see here on the left. And we sent this first draft of the TPP out internationally for a Delphi survey, which is actually just now closed, to try and get further input and insight into the recommendations that we've developed, so that at the end of this month, we can discuss them at a virtual symposium, which will bring together people with CF, clinical teams, it will bring together companies that are working in the space and other key stakeholders to really discuss and debate the content of the TPPs and to generate some consensus so that we can finalise and publish these documents and disseminate them widely, with the idea that we can start to spark new drug discovery efforts in the area and strengthen existing efforts to start bringing new treatments to people with CF sooner. And just to give you an idea of what this means to people with CF, Angelo said that being involved in this focus group was really helpful to him as a patient. Lorna, who has a child with CF, says that she hopes that these CPPs can influence the development of future treatments so that they can be easier to manage and more tolerable without loss of efficacy. So hopefully this shows you a new approach that we're taking to try and create guidance for the community to really catalyse new efforts and strengthen existing efforts in a very specific disease area. So we've been in progress for about 1.5 years now, and just to kind of talk about what we've achieved to date through this initiative, we've brought together a core group of leading experts who are committed to our mission. We've mapped, engaged and connected within the sector. We've leveraged academic and industry expertise to drive impactful programs of research, and we've involved people with CF in everything that we do. And moving forwards, we'll continue to build these resources for the community. We're going to build on the engagements we've made with the community to create a unique cross-sector network. And we're going to be looking to identify and support promising CF antimicrobial programs to really help them to reach the clinic sooner. So I'm just going to end there with my acknowledgements. There's a lot of people on this list, but mostly to my colleagues at the Catapult and the Cystic Fibrosis Trust, especially Paula, who I work with extremely closely through this initiative, and to the members of the CF Syndicate and AMR Steering Committee, and to the contributors of our various programs. So thank you very much.
Thanks very much for a really interesting presentation and for keeping to time, Jessica. So next I would like to introduce Cara Lepore, who is from the Pew Charitable Trusts. Thank you.
Thanks so much, Jessica. Good morning, good afternoon, everyone. My name is Cara Lepore. I'm a senior associate for the Antibiotic Resistance Project at the Pew Charitable Trust. Thanks so much to the organizers at JPIAMR and the UK Medical Research Council for the opportunity to present today. And I'd like to follow up on the great conversation in the last session about choosing products to develop with a current overview of the clinical pipeline. So if you're not familiar, the Pew Charitable Trust is a global nonprofit. Our mission is to provide data and analysis to inform public policy on a broad portfolio of issues. One of those being our antibiotic resistance project, which works on antibiotic stewardship and advancing policies that spur the development of new antibiotics. So some of you may be aware of our team's assessments of the global clinical pipeline of antibiotics and non-traditional products to treat serious bacterial infections. We've been reporting on this data since 2014. So the scope of this pipeline project reflects the need for a diverse portfolio of effective medicines that treat the drug-resistant bacterial infections today and tomorrow. So we identify both the small molecule antibiotics and innovative non-traditional products, including things like vaccines, antibodies, bacteriophages, et cetera, which could help protect patients from bacterial infections in new ways, or maybe used in combination with traditional antibiotics. And as far as the scope of these pipelines, we include systemically acting drugs with a new chemical entity that treats serious or life-threatening bacterial infections. And we report on each drug's current development information, for example, their company and development phase, drug class, mechanism of action, including novelty for small molecule antibiotics, activity against escape pathogens, CDC urgent, WHO critical threat pathogens, and the potential indications that the candidate will aim to treat, including QIDP or Qualified Infectious Disease Product designations, which is a US-based designation, if applicable. So first I'll give a brief overview from our most recent small molecule antibiotic pipeline update. So looking at the pipeline for traditional small molecule antibiotics, our message has remained consistent over the past now 7 years. And that's that there are not enough antibiotics in development that target the most dangerous pathogens, and not enough are novel classes. Unfortunately, still remains true with this latest pipeline update. So there are 43 small molecule antibiotics in development, broken down in this graph by development phase. And against the highest threat pathogens deemed by the WHO and CDC, there are 24 candidates that do have expected activity, as you see on this right graphic here. And when we say yes for activity against these, the CDC, urgent, and WHO critical threat pathogens, that indicates a drug that has convincing in vitro and or in vivo data showing activity against these corresponding bacteria. So the majority of these highest priority pathogens, many of you know, overlap on the WHO and CDC lists. The other pathogens, C. diff, gonorrhea, ESBL producing Enterobacteriaceae, are acknowledged by both of those lists in some capacity as well. We also break out these 43 candidates by novelty. And when we're talking about a candidate that has a novel class, we mean that that has a core chemical structure that has not been previously used as a systemic antibacterial drug in humans. Novel drugs are an important part of the pipeline because they're less likely to have existing cross-resistance to them. But as we know, they're very difficult to discover and develop. So breaking out the current pipeline by novelty, 10 of the candidates on the pipeline do consist of novel classes. The bad news is that none of those 10 can treat gram-negative escape pathogens. They do address gonorrhea, C. diff, other gram-positive infections. So even though there are 43 products in the pipeline, very few actually have the characteristics that we need to combat antibiotic resistance. Only about half of those to treat the WHO or CDC critical pathogens, and a quarter novel classes. And on top of that, as has been discussed, an average of 80% of infectious disease products that enter clinical testing will ultimately fail. So this kind of paints a picture of the urgent need to reinvigorate the pipeline of antibiotics in development. To give a more in-depth view of the state of the pipeline, we've also taken a look at the companies that are developing these clinical pipeline drugs. So the graphs here are looking at the shift to primarily pre-revenue companies, even over the course of the last 7 years. So when we talk about a pre-revenue company, we're looking at a company that does not have any previous experience with actually bringing a new drug of any therapeutic area through regulatory approval and commercialization. And as you can see, the shift from 46 to 73% being pre-revenue companies also correlates with the big pharma companies that have left the antibiotic field. So only 2 of the companies with an active clinical antibiotic in 2020 was one of these, was a top 50 pharma company, which is down from 8 in 2014. And we've also unfortunately seen bankruptcies from the smaller biotech companies that have remained in the antibiotic space. In fact, actually 5 of the 13 FDA-approved antibiotics since 2014 were brought to market by a company that has since divested its program, filed for bankruptcy, or was sold for far below its evaluation. So, you know, these are companies that have brought successful new drugs to market and then they struggle to survive after. So that further emphasizes that the antibiotic market is broken. And to give a brief overview of our assessment of non-traditional products. So we evaluate the non-traditional pipeline because while traditional antibiotic innovation is essential to combating antibiotic resistance, we also need these kinds of outside-the-box approaches. Many of these products that we've captured are unlikely to fully substitute or replace antibiotic use. Rather, they'll be used more in conjunction to help disarm the pathogens or as prevention. These products are also usually active against a limited range of pathogens, rather than antibiotics, which are more likely to be broad spectrum. So the majority of the non-traditional products we captured in the pipeline are well-known therapeutics, such as vaccines and antibodies, which have successful track records, obviously, in treating other types of diseases. But we also look at entirely new types of therapies that have never been approved for use previously in human medicine. For example, lysins, antibiotic inactivators, and viral inhibitors. And as we know, all of these innovative approaches are exciting possible new tools. They are largely unproven, so the road ahead to successfully prove safety and efficacy especially in combination with an antibiotic, is challenging. So we have a number of pipeline resources on our website that we hope will be helpful for your use. We have interactive analyses of both pipelines, as well as downloadable Excel sheets of the data. We have summary pieces to help distill the data from our updates. And since we've been tracking the pipeline since 2014, we also have an interactive tool that tracks the movement in the pipeline over the last now 7 years. And I've included this link at the bottom of the slide if you'd like to take a closer look at these resources. Most of you are also probably aware that the WHO also carries out an annual antibiotic clinical pipeline review with a lot of additional excellent resources. And they just released their annual update of the clinical and preclinical pipeline last week. We have slightly different methodology, but we coordinate our analyses to make sure we're providing the most accurate and up-to-date information to the community. So our messaging is also always very aligned. And because of this, last summer, we released a joint call to action to reinforce the urgent need for development of new antibiotics. So directing this call to policymakers, pharmaceutical companies, funders, other stakeholders, we urged increased public funding for early stage research, increasing support for push and pull incentives, including public-private partnerships like GARB-X and GARB-P, and identifying innovative solutions for sufficient return on investment for new antibiotics, while obviously ensuring their appropriate use. So these are solutions that have to be sustainable and robust, obviously, to revitalize the antibiotic market long-term, while also prioritizing stewardship to effectively combat antibiotic resistance. And I'd like to thank the members of my team at Pew, as well as our external pipeline experts for supporting this work. Thanks so much for your attention. I'm happy to take questions following the session.
Thank you very much, Cara. Again, really interesting presentation and perfect timing. So next, I would like to introduce Rosemary Dorrington. So she is from Rhodes University and is leading a South Africa UK antimicrobial drug discovery hub.
Before I start, I just want to reach out to our colleagues at the University of Cape Town who on Sunday and Monday suffered the devastation of a runaway fire on the mountain above the university, and there's been extensive damage. And to all of you guys, we really, you have our sympathy, and anything we can do to help, you need to call on us. So I'm going to speak about a new initiative that is funded by the Newton Fund and the South African Department of Health through our respective medical research councils to develop capacity for antimicrobial drug discovery in a joint initiative between the UK and South Africa. And this particular initiative is focused on exploiting existing capacity within the consortium for natural products research as a pipeline for developing compounds for the antimicrobial discovery pipe. I'm sorry, yeah, to progress into the antimicrobial discovery pipeline. So the aim of the hub is to develop a cutting-edge research platform for antimicrobial discovery, and also to establish the infrastructure and human capacity to provide support for future drug discovery efforts. Our objectives are to establish a discovery pipeline of natural products and synthetic compounds, and to assemble these into curated libraries that will be made available for screening to other drug discovery efforts, and also accompany these curated libraries with a database of known compounds and activities. There will be a strong focus on the development of an omics platform with capacity for in silico mining of genomes and metabolomes. And in doing this, we hope that we will be able to supply novel hit compounds that can be progressed into lead optimization in a second phase of the project. In parallel to this, the hub aims to develop research networks that can harness existing compound libraries and extract libraries that can be screened through the platform. So I had to put this in. This is an image we took a couple of weeks ago in Algoa Bay, which is an hour's drive from Rhodes University. It's our laboratory, marine laboratory, and it's a reef approximately half a kilometer offshore. And it's put specifically to illustrate the extraordinary biodiversity that, in particular in South Africa, both terrestrial and marine, much of which is unexplored, and which supports the discovery of novel compounds of natural from the biological novelty that we find in our environment. So the advantages of natural products are that they have unprecedented chemical diversity and structural diversity, and that their structures have evolved over time with their targets to be highly efficient and refined. They are highly selective for their targets and extremely potent inhibitors of their biological targets. And the biosynthetic pathways of these compounds is overwhelmingly of microbial origin. So for many of the natural products, with the exception of the several plant natural products, but in terms of certainly marine natural products, the production is by symbiotic or microbes in the environment. The disadvantages are well known, and until recently have provided some discomfort about pursuing natural products as sources of chemical novelty. These include the problem of supply. It's often difficult to get compounds in sufficient quantities to do much work with. You have to have ways of screening complex extracts that can contain up to 20 or 30 different compounds at different concentrations, and you have your assays have to be able to pick up activities that you're looking for. It's difficult to often difficult to dereplicate the active compounds in the extracts and to identify the active compounds, because there is often synergy in some cases. And finally, these are complex molecules and they are frequently difficult to synthesize in sufficient quantities. But we now have new technologies. We're able to work in much smaller quantities, and we are able to use these new technologies to progress natural products far more rapidly than, say, for example, 10 years ago. So in phase one of this project, we're really looking to mobilize a diverse pipeline of natural products from plants, existing plant natural products collections, marine natural product libraries, existing and new, and then to mobilize microbial natural products or potentially active compounds from existing fungal and microbial collections held within the consortium. And in parallel, to mine these, particularly the microbial collections, for novelty in terms of biosynthetic pathways for new chemical entities. These extracts will then be are in the process of being mobilized into new chemical extract libraries that are being screened through a bioassay platform that we're developing within the consortium. This is in collaboration with H3D, who have an escape high throughput screening platform at the University of Cape Town, and then our own in-house more low throughput screening platform that we're using to screen the natural products extracts. We have a research platform in place for supporting the dereplication of the natural products, extracts, and identification of active compounds, and then progressing those for ADMET and ultimately prioritization for phase 2 and further characterization to optimize the hits and potentially progress to lead optimization. So the consortium comprises 12 institutions, and including 7 South African and 5 UK institutions, with broad expertise across the different activities of the platform. And I'm not really going to go through them, but members of the consortium pulling these expertise together, we feel provides us with the opportunity to develop the capacity for early stage discovery in the of potentially active novel natural product compounds. And then I did want to put this in briefly. It's taken some time to get started. The project was initiated in June last year, and we've had all kinds of problems with getting started, considering we've been in lockdown since March. But we have been able to start doing some screening, and currently we've screened approximately 800 pure compounds of natural products and medicinal chemistry origin. They've been screened for escape activity, and we're now following up active compounds with activity against Staph aureus, Pseudomonas, and Acinetobacter. And we're using E. coli as a ready screen to begin to prioritize compounds for further work. And I just wanted to put this in to illustrate the importance of keeping old chemistry libraries available and being able to rescreen them. So we did a pilot project in which we screened approximately 250 chemical extracts that were collected 25 years ago from marine invertebrates, mostly sponges, collected off the Southern African coast. The screening has returned 6 hits with potent activity against E. coli and Staph aureus. And I just want to illustrate why one sometimes gets really lucky. So for example, as a proof of concept, the 2 E. coli hits, these 2 sponges over here, turn out to be the same species, but collected at 2 different times, one in Mozambique and one off the South African coast. And on the right here is a sponge from which we recovered compounds that were a family of compounds that were identified 10 years ago as having specific activity and against methicillin-resistant Staph aureus. We recovered not only the compounds that were identified previously, but we've identified new compounds that appear to have broader activity against other members of the escape pathogens. So there's real value in curating and looking out at screening as new assays become available to look at screening old libraries, particularly chemical extract libraries where you could be screening 20 or 30 compounds in a single extract. Excellent. Can you wrap up now, please, Rosie? Yes. So the project is funded, as I said before, through the Newton Fund on the UK side, by the Newton Fund and the Department of Health. But I also want to point out these funders at the bottom. You're only as good as the access you have to novel biota, and we are very fortunate to be able to, supported by the British Antarctic Survey, the South African National Research Foundation, and projects that enable us to get into the field and sample novel environments. Super. Thank you. Thank you very much, Rosemary. That was really, really interesting. So the last speaker in this session is Peter Jackson. So he will be speaking on behalf of the ICON Consortium, so Infection Innovation Consortium. So Peter, the floor is yours.
That's great. Okay, well, Jess and the team, thank you for the invitation to speak this afternoon. What I want to do is to talk about a superb case study where collaboration is essential to making progress, and that's the ICON Infection Innovation Consortium. I'm Pete Jackson. I'm Chief Exec of Infex Therapeutics, which was formerly known as the AMR Centre. I'm on the exec board of ICON. I'm also a member of the project advisory group for the pull incentive that's being run by NHS England and NICE, the trial project. And I'm a senior advisor to Milken and Fasticures in DC as well. So what is ICON? So there's essentially the northwest of England. There's a network of partners that in the past had had significant bilateral relationships, and we realised that we could achieve an awful lot more by knitting those together into a formal consortium. And so we did that and applied to the UK UK government Strengthening Places Fund, and were successfully awarded at the end of last year £19 million from UKRI. At the time, for an overall project of 119 million, so with 114 million with 95 million of private sector leverage. Now we've grown that to 147 million of private leverage, so that the total program at the moment is 173 million, so well over $200 million to deploy into this area in the next 5 years. So it's led by Janet Hemingway from the Liverpool School of Tropical Medicine, and the partners include University of Liverpool. I know many of you will know William Hope and his expert group on PKPD. The Evotech group at Alderley Park, specialising in microbial analytical and testing methods. Global major public health company Unilever, they have a strong interest in the public health and innovation elements of the AMR agenda. Ourselves, of course, and the Royal Liverpool and Broad Green University Hospital and within the UK NHS, and in particular Richard Fitzgerald's phase 1 clinical trial unit there, which specialises in infectious disease and respiratory disease. But it's an open partnership, so we would very much welcome others who would like to join, particularly who can bring funds and expertise. Our ambition is to build a £1 billion initiative over the next 10 years, so $1.3 billion to invest into new infection translation. So our aim is to become that first choice for anti-infectives translational research and development by partnering with particularly SMEs, research institutions, other academic organisations, and large-scale industry, big pharma. And we do that by creating capacity and offering the ability to co-develop and de-risk across infectious disease, drugs, and diagnostics. So we have diagnostics platforms as well, and connect that to the problems worth solving that John, Rex, and his panel talked about earlier. So how do you build a target product profile that meets patient and actually global payer needs? And we really need to develop cheaper, quicker, smarter pathways, and a lot of that is also by having really good dialogue with regulators. So, for example, taking advantage of the new UK ILAP, the innovative licensing and access pathways, where we can have those discussions with regulators earlier rather than later. And all in all, it's about this translational piece to progress products from discovery through to use in patients. So there are a number of platforms within the ICON initiative, and just talking about the ones that are more relevant to new products for AMR. So building on what Rosie was just saying, we have a natural products platform, hits to leads, which I'll say a bit more about, which our aim there is to support a number of programs to get at least 2 new therapies into clinical trials and developing new in vivo models. So I think really good opportunities for collaboration here. So new models for Acinetobacter and nosocomial pneumonia infections. And then on the public health side, actually, what are the drivers for AMR? So what much more research needs to be done in being able to predict transmission and infection from resistant infections. So just to fill in on the Hits to Leads platform, Infex is taking the lead on this particular part of the initiative, and it's really about bridging this preclinical valley of death. Incredibly difficult for early stage companies to raise private funding in this area. We have capacity up to 45 FTEs across microbiology and medicinal chemistry. Most of the team based at Alderley Park, which is AstraZeneca's former research headquarters, but also on site under umbrella agreements, we have access to all of the preclinical service providers that you would want to put together a successful program. And then moving on, after we've generated a program that moves into lead op and preclinical development, that's where the wider ICON partnership swings into place. And as I mentioned before, ultimately with the ability to put programs through phase 1 at the Royal Liverpool Hospital. So we've been going for around 4 months now, 4 or 5 months, and we got the first projects underway with microbiotics from just outside Boston in the US, and a number of programs from LifeArc, a leading UK philanthropic-backed research centre, as well as other philanthropic organisations and universities that are not in the public domain right now. So just summarising, ICON is open for partnerships, and that's whether you're in a research institute, in academia, whether you're in industry, or you have a philanthropic interest, and we're open to partnerships from research all the way through to clinical proof of concept. So we think it's a major initiative backed by the UK government and the private sector, and we would very much invite you to join us to rebuild the anti-infectives pipeline. And the contact details for ICON and Infex are there on the bottom of the screen. So thank you.
Great, thank you very much, Peter. So if the rest of the panel members wouldn't mind putting your cameras back on, the team will sort of arrange us, and we've got 5 minutes for questions, so that's done quite well, I think, for presentations. I'm going to start the questions off with a question for Jess. So what is the best way, do you think, to engage patients early in the process? So you made the point that making products that are relevant to the patients was important. How do you get in touch with them? How do you liaise with them?
I think the thing that we've really benefited from is working very closely with the Cystic Fibrosis Trust. So obviously medical research charities have that close engagement with the patient population. So we've been able to engage people through membership of our steering committee. So they've been involved in the strategy of the syndicate from the outset, but also through focus groups and other ways. So I would really encourage people, if they want to engage with patient populations, to get in touch with the relevant medical research charity in the first instance, probably.
Okay, thank you very much. Second question, Rosie, is directed for you. So it's come from the audience, the attendees. In connection to the South African UK project, are there networks set up to investigate the mechanism of action of these compounds, either in vitro or in vivo?
We're in the discovery phase. We're funded for phase 1. And as the compounds that we think are going to be prioritized for further hit optimization are identified, we would certainly be looking to establish networks, yes, for phase 2 of the project. But if anybody's interested, we'd really welcome talking to them.
Okay, thank you very much. Cara, a question for you from me at this point. Where's the greatest, where do you see the greatest area of anti-infectives therapeutics in terms of the fluctuation over the last 5 years? So where have you seen the most sort of interest in the pipeline over the last 5 years?
Yeah, that's a good question. Honestly, the last 5 years has been pretty consistent in the clinical pipeline. I would definitely recommend looking at the WHO's excellent analysis of the preclinical pipeline, as was discussed in the last session as well. There's a lot of really exciting novel therapeutics that are in the preclinical pipeline that we hope will reach the clinical pipeline and kind of alter that the focus more towards the critical pathogens, the gram-negative pathogens, etc.
Okay, great. And Peter, there's a question that's come in again from the audience for you. Is ICON working as a service provider to support early antibiotics discovery projects, or do you only undertake projects that you deem meet criteria for funding?
Yeah, so that's both is the answer to that. So we have, at the moment, we don't have unlimited funds, so we have to have some sort of selection and triage, but certain elements within the overall consortium are offering services. Evotech, for example, are a well-known service provider, and William Hope's team at Liverpool, I think, are open for paid services in PKPD. But what I would say is that any paid services are significantly subsidised by the consortium funding, and in particular the access to phase 1 capability would be at a significant discount, for example, to that compared with an external CRO provider.
Okay, thank you, Peter. And I have one more question that I'm going to direct to Rosie. So Rosie, what do you see as the most important aspect of the transnational collaboration that you've established? What benefit does that bring to you?
When we started the project, we anticipated that there would be a great deal of exchange between institutions in the UK and South Africa, and that there would be a strong training and skills development component. But we've learned that there are other ways to pool resources. And we've also, I think, is a problem. But I also think that one way that it's not a waste of time, but a lot of energy is extended to traveling backwards and forwards between different laboratories. And we've found that we can develop the skills in our home laboratories much more quickly by being more focused. The other thing I think is really important is being exposed to, for us in South Africa, is being exposed to the next phase of expertise, as was mentioned in the question, discovery biology, and taking compounds that have activity and progressing them through to hit optimization and lead progression, which is capacity we really don't have on our side.
Excellent. I'd like to thank you all very much for a very interesting session, and it's time to hand back over to the workshop chair, Marc Lemonnier. Thank you.
Thank you very much, and I really want to thank all the speakers, the moderators, all the participants, because it's been a very, very interesting introductory day to this conference. We've heard a lot of things, a lot of very important things, and from a very diverse group of people, covering, I think, all the aspects of this problem that we have with needing to have a sustainable pipeline of novel drugs and that go to patients and that are used appropriately. So we've tried to summarize a little bit some of the hot ideas that we've heard today I don't know if anyone can maybe share the slide deck, because I don't have it with me. And I have to say, this is amazingly difficult to set up a workshop from a distance. We're all used to Zoom, and with COVID, this has become part of our landscape, but it's still extremely difficult, and I'm really impressed by the organizers. So thank you very much for your support. So session one, I think, was extremely important in order to set the scene. And we've seen that, yes, the priority pathogen list is still valid. And as we've seen, shown by WHO, by also the Pew Trust and others, the products in the pipeline are insufficient to tackle the problem. And what we see also is that really most of what we see in the pipelines rely on combinations of traditional antibiotics or the improvement of existing antimicrobials. And it's true that we think of a global problem, but the resistance is variable according to different geographies. So we'll need a complementary set of solutions to tackle that problem. The magic bullet, we all want it, and as John Rex said, there's a number of things we all want to see, but in reality, I think diversity of approaches are going to be needed in order to tackle that problem. There's a number of difficulties. We've seen, yes, clinical trials is difficult, especially in geographies where resistance is still low. And those strategies sometimes, well, it's true that there's a problem of balance between geographies, because in high-income countries, sometimes it's easier to develop some of these precision therapies, and therefore there's a difference. And GARD-P, for example, has shown that they take a different approach. They acknowledge that low MIC, LMIC countries have a pressing need that needs to be addressed. So it's really interesting, because we always think of a global uniform problem, and actually it isn't. And I think that one of the biggest, the main ideas we've heard today is that action and dialogue are needed. Dialogue between very different stakeholders. Payers should talk with regulators. Academics should talk with the industry. Private capital needs to get something from public funding from the governments in terms of pool incentives. So there's we're all in the same boat, and we need to align our objectives. This is, I think, one of the key things. The challenges are different in animal and human health. That's true. What we've seen with big pharma leaving the space is that there's a gap to fill, and universities cannot fill that gap. Universities have and I'm saying academic research, plays a major role in here in terms of generating the science that will and should become a product. But what we are seeing now is that SMEs, small companies, pre-revenue companies, as the Pew Trust puts it, they're bearing the bulk of innovation. It's more than 80% of the preclinical and clinical pipeline. And often in those small companies, you have ex-pharma people, so there's a lot of expertise there. But SMEs, they need to talk to pharma and need to talk to academia. Grant and funding shaping the drug discovery process, yes, but grants and funding are listening to what WHO, CDC, and other organizations that are really showing the picture of reality. So saying these are the trends, these are the resistance trends, and these are the gaps. The money, the public money, the pool incentives, they will be shaped by reality. And of course, reality is not written in stone, because in 10 days, maybe the resistance will be different to what we see now, and therefore the priorities will change. But one thing is sure, the science and the projects will be funded looking at those targets. So any science that is now being kicked off should bear in mind this. It's absolutely important to understand where the targets are in order to, get on with the science. Next slide, please. Yeah, so then the session 2 also very interesting. Of course, a lot of what I've said was also said during session 2, but there's the concept of clinical need and societal value. This is very important. I think the only thing you can measure is an individual benefit when you want to show demonstrate proof of concept in the clinic. However, here we're talking, and COVID is another example, of course, there's a benefit to those around me by doing certain things, by delivering some interventions and new treatments. This benefit is not only to me, it's to those around me. Or as of today, this is extremely difficult to assess. And Marco Cavalieri from the EMA said he's very conscious about this, but the regulators are now open to finding a solution. And that's not only a problem of the regulators acknowledging the problem, it's having the tools to measure the collective benefit. And that is also very important when you're developing non-traditional products. You need to be able to identify those novel measures of value of your product that are outside the standard way of measuring efficacy and utility in the clinic. So there's a lot of work to do, and also that will help convince payers that this needs to be paid differently. How do you value a product if you don't have the data to say, well, actually, this is where the benefit is, the target population, right? So there needs to be other ways also to show the value for those products. And then, of course, pool incentives, we need them. There is a lot of things that need to be done. The duration of the drug development is extremely long. So, of course, we're pressed by time. It's time to act, and it's time to identify those new solutions. And finally, next slide. Day with session 2A, and here we had very interesting talks from a number of initiatives that are supporting and giving the right tools for those approaches to develop the new drugs to patients. And we've heard from the CF syndicate in AMR that involving patients is extremely important, not only for CF, but to understand the patient's need and to develop tools. They can be depository or they can be new target product profile. It's extremely important. We heard again by the Pew Trust the confirmation that the antibiotic pipeline is inadequate, and that, of course, novel drugs are not less. They're more important. They're very important, but they're difficult to develop. Attrition is there. Natural products can bring also interesting solutions to the table. They remain relatively unexplored, diverse, selective. They come, of course, with their own baggage in terms of challenges for development, but there's value in screening all libraries, and there's also very interesting initiatives to bridge the preclinical valley of death, as Pete Jackson showed with the ICON initiative. So thank you very much, everyone.